Hi everyone, my name is Varun Ramraj and I'm going to be conducting your lecture today on information, networks, and uses. Okay. Now before we get into this, I just want to clarify that these terms are very broad: information, networks, and uses, and we're going to spend some time defining what these terms mean. More specifically, however, we're going to define them in the context of bioinformatics. So the lecture today is going to focus on a survey of computing resources and challenges that bioinformaticians face. Bioinformaticians like you who will face some of these challenges in this course, even before you get out into the real world and look at bigger problems. Now, this is my email address, so it's varun_in_struct@zoho.com. Please do not hesitate to email me at any time with any questions or comments or concerns you may have, and I'm more than happy to get back to you as soon as I can. I am based in the UK, so just factor that in, that there is a time difference, but I will endeavor to get back to you as quickly as possible. Right, let's get started. Now, before we dive into it, I like to put up an overview slide just to focus our discussion today and to make sure that we get through the following points. So, right off the bat, I want to discuss some lecture goals with you. Okay? Now, secondly, I want us to spend a little bit of time dissecting the title. So I think this is very important. When you see the title of a lecture during this course, spend a little bit of time dissecting it to understand exactly what it is you're going to be covering, because that's going to focus your mind into the subject at hand. Thirdly, we're going to look at the anatomy of a network. What is a network? What does it look like? 4, we're going to then look a little bit at some bioinformatics data. So what does bioinformatics data look like? What kind of data do bioinformaticians face every day when they go to work? Now, fifthly and sixthly, these are two very important concepts in bioinformatics that are pipelines and databases, okay? Now, we're going to look at these in the context of networking as much as possible. But we will look at a broad overview of these before we get into it. And then finally, before we finish, we're going to look a little bit at manipulating bioinformatics data. And finally, we end with a summary of what we've covered. Before formally we get started, I need to tell you a little bit about me, of course. So I did my undergraduate and my master's at the University of British Columbia in Vancouver. My undergrad was in biology and computer science combined, and my master's was in bioinformatics proper. And I've just finished my doctorate here at Oxford. It's a DPhil in clinical medicine, but I've been spending my time building bioinformatics software tools for studying large-scale research problems. And with my computer science background, I'm very interested in building high-quality software. Okay, so these are my research interests. Feel free to ask me via email at any point if you're interested in more of my research. We can certainly have a discussion about this. Right, let's get started. So right off the bat, don't worry. This lecture is an introduction, but by the end of it, you should be able to do a few basic things. You should understand what a computer network is. Okay? The term network gets thrown around a lot. We use it every day in our lives. But to properly understand what it is, and in the context of bioinformatics, that is one of my lecture goals, for you today. Secondly, I want you to understand what a bioinformatics pipeline is. Thirdly, I want us to spend a little bit of time looking at bioinformatics data. We're not going to get into it too deeply here. You're going to have many labs and tutorials to look a little bit more in depth at this data, but we do want to go over a brief overview. Fourth, be able to name some of the popular data repositories, so places where bioinformatics data is stored and is available to you. And these are repositories or databases or websites that you will be using repeatedly in this course. So being able to name some of them today is one of my goals for you. And finally, this is absolutely the most important take-home point today, okay? I want you to gain an appreciation for the sizes of the data sets that we talk about in bioinformatics. Bioinformatics is special because of the size of data that we work with. And it's not just simply a matter of using standard software packages to work with this data. You need to make some key decisions and you need to be able to estimate the size of data that you will be working with based on the networking resources. Again, remember this lecture is about networking. So this is a very, very key skill, and we're going to look at an example or two about this today. So let's get started. Dissecting the title: Information, Networks, and Uses. What is information? In bioinformatics, we are interested in text-encoded genes or protein datasets. This is the information we look at. Okay, so pretty much all the data that you will experience and you will encounter as a bioinformatician is going to be text-based, and most of those datasets are going to be genetic data or proteomic data. Okay, and this is the data that comes in as your input, and you get to manipulate it, you get to conduct studies on it, you get to do some statistics on it, and you return a result to the user. But primarily we are interested in text-encoded genes or proteins, plain text. Networks and uses. And this is a very simple definition that pertains to this class. Okay, so don't worry about getting too in-depth into this, because networking is a huge topic that is a course unto itself. But very simply, a network, as I define it for this course, is a set of interconnected machines. That's all it is. It is a machine that is connected to another machine that is connected to another one. And each machine has a discrete job. So each component of this network is doing a discrete job or a set of jobs. Okay? And a network has an input and an output depending on what I like to call the use case. That is the reason for this network to exist. By the way, I do operate on a wireless headset, and occasionally it does try to disconnect. So if that happens, I will reconnect right away. So my apologies in advance for that. Let's take a quick example of an office network, okay? Each office worker has a computer that connects to an email server, a file server, a network printer. Think of your own university. When you're sitting in the computer lab, you have all these components that your computer is connected to. So in this network, the computer, email server, file server, the printer, all these things are the discrete machines that I have described above. And these machines talk to each other. So here's a simplified diagram of what I just described. So you've got your individual computers here. They're connected together by a switch that then goes to a server that maybe controls a printer. So if you send a print job, this is what it does. It goes up to the printer this way. The server is then connected to a router that then connects other servers, and eventually it all leads out into the internet. Okay, so think of this diagram up to the internet point here as being a simplification of your university network. And this is what a network looks like. So let's dissect this a little bit further. So what components, what properties does a generic network have? So let's abstract away from offices. Let's abstract away from bioinformatics and all these specific use cases. Let's look at it generically. A network has something called a topology. It is a property of a network that describes each machine, or node more generally. Okay, this is an important term that you will encounter repeatedly in bioinformatics. So a network topology describes each machine or each node and how it is connected to the other nodes. So it answers the following questions. What does a particular node do? And what is that node's input and output? A network also has a geographical layout that is different from a topology. The geographical layout describes geographically, physically, where each machine or node is located. So you could have a machine that's located in computer lab A. You could have a node that's located, that's a printer perhaps, that's located in a central office, somewhere else on campus. So that description of the location is what the geographical layout is telling you. Now, why are these two components important? They both affect the speed and the ability to store things on the network. Okay? Speed, storage, and bioinformatics. These two things are mission critical in bioinformatics because the data we work with are huge. Now, I put this in quotation marks because we're going to define this later. So when you're working with bioinformatics data, keep this in mind, okay? So when you work in the labs in this course, if you have to download a big genome file of the human genome, for instance, if it's stored in Trinidad, if it's stored at the university, for instance, it's going to be faster than downloading it from the United Kingdom. And oftentimes, data repositories will give you options for where to download from. And in those cases, the general rule, that can be broken sometimes, but generally, pick the node that is pick the download location that is closest to you. Okay? So, understanding bioinformatics data. So we've seen a little bit about the network, so now let's look at the data that goes through the network. All right? Most data sets that we work with are stored as human-readable plain text files. These are not Word files. They're not even rich text. They're not PowerPoint. They're none of these things. Simple, plain text files. You're going to see why later, but as a hint, it's got something to do with the size of the data. Even before bioinformatics was a formal science, computer science has always had various file format standards. This is to enable software to read and analyze data in an agreed upon format. These formats are called standards, and in bioinformatics we have many standards for the kinds of data we work with. The input that you work with in bioinformatics is usually sequence or structure-based input. Again, the input data is presented as a plain text file, but it contains information on sequence or structure, and going back from the previous slide, it's usually genes or proteins. So the software packages that we use in bioinformatics are designed to work with various file formats sometimes. So we have old formats that older files are stored in, so files that were created in the 70s and 80s have a particular format. Maybe that format is decided to be changed for some reason. Software packages will often allow you to use the old formats, okay? So this is an important concept in bioinformatics because of so many different file formats that have been evolving over the years. These software packages will often provide options for input and output file formats, so you can change based on the kind of input and kind of output you want. This is very useful. So remember, if you're working with some obscure file format, chances are the software packages that you're using will allow you to import or export that data. As an example, the most basic format and the most common one that you're probably going to be working with, at least in this course, is the FASTA format. FASTA is an acronym. It's based on a software package, a set of software tools for bioinformatics back in the day. This is quite an old software suite, but the file format that they used was called the FASTA file format, and it is the most popular format that is being used today. It is the most common way of storing sequence, so either DNA, RNA, or protein sequences, generally. It consists of a header line, which starts with this greater than character, followed by some information or metadata, another key bioinformatics term, metadata about the sequence. And then it has the sequence itself. So let's take a look at an example. Here's a FASTA file. Here's your greater than sign. There's some information, some numbers, maybe some accession numbers or some IDs for this sequence. Here is some information about what this is, and here's some more information perhaps of which species it came from. So in fact, this protein sequence, it is a protein sequence. You can tell by the letters it's not A, T, C, and G, so we know it's a protein sequence essentially. Each letter is an amino acid. This is a protein sequence for the cytochrome B protein that's taken from an Asian elephant. That's what Elephas maximus is, a scientific term for an Asian elephant. GI and GB identifiers, don't worry about it for now, but they're basically unique IDs for you to be able to find the sequence in various databases. So one-letter codes, okay, just a quick side note here. As you go through this course, you're going to probably find it very useful to keep a reference of one-letter and three-letter codes for DNA, RNA, and proteins with you, just because we're going to be looking at this, this format many times. But just think to yourself, why don't we spell out each residue in full? Why don't we say lysine, methionine, arginine. Why don't we say that? Why are we using one-letter codes? Is it just because it looks nice, or is there some other deeper reason? We have discussed this, so think about this for a second. Now let's move on to pipelines. So bioinformatics pipelines are going to be very common units of software that you use in this course. So remember that networks are they consist of interconnected machines. I've mentioned this term to you. So in bioinformatics, what is this machine? What information does this machine hold? What can it do? The machine in bioinformatics is called a pipeline. A pipeline is usually consisting of one or more software packages that are connected together to process a particular kind of data. And the reason this is relevant to networking is because different pieces of software can exist on different machines, on different computers, maybe in different parts of the world. But they can in fact be connected through the same network. So in your own university, think of a case where you are sitting in the computer lab and you are using a piece of bioinformatics software that sits on a different machine in a specific bioinformatics lab, for instance. That's what we're talking about. The pipeline is between your computer and this other machine that sits somewhere else. Let's take a very simple case study for DNA sequencing, because this is something that you will be learning about in more detail later. But basically, you have a DNA sequencing machine that sits in a wet lab somewhere. You have scientists in white lab coats, usually. Sometimes they're colorful. But generally white lab coats going around and doing chemistry on these DNA sequencing machines to find the sequence of something important, say a new bacterium that's causing some kind of epidemic. The raw sequence from the machine is sent to a bioinformatics pipeline. Okay, so again, the pipeline is a set of software. Not necessarily, it could be just one package. We still call it a pipeline. Okay, the pipeline maybe is in charge of looking at the quality of the sequence that's come out of the sequencer. Maybe this DNA is badly damaged due to radiation. Okay, so these are quality control mechanisms that the pipeline is able to do. And maybe it's looking for genetic mutations that could cause a particular cancer. And each of these components is handled by a different software package. So these software packages are connected together to form this pipeline. But the network here includes the DNA sequencers themselves, and those sequencers are nowadays connected to the internet, to your network. They send the data through the network after they perform their chemical reactions in the lab, and then they send this data to the bioinformatics pipeline. As far as the pipeline itself, as I mentioned before, it could be split across multiple machines. Maybe you have a compute cluster that has a lot of very powerful machines that can analyze the data quickly. But compute clusters generally don't have very big hard drives. And remember, we said we're working with lots of data. So maybe you have a file server or a storage unit which has a lot of very big hard disks where the cluster can then dump its output. So the pipeline is split across multiple machines that are connected through this same network. Now let's look at databases. Now, pipelines are one very important component. Databases are the other very important component in bioinformatics. So why do we need them? A database, very simply, is designed to store very large amounts of data in a quickly searchable and retrievable manner. This is the key. You could have a file server which has hundreds of thousands of files, but it's like having a filing cabinet where you just throw in a bunch of papers. You need to have some sensible way of storing them and being able to find them later. And that's what the database allows you to do for the data that we work with. A database is often curated, another important term in bioinformatics. Generally, I have been highlighting important terms for you to know in bold. Okay? Curated means that dedicated people maintain the quality of the database. So sometimes in experiments you get very poor quality. Okay? And it's the job of the bioinformaticians to then go in and see and actually assess and put a number to the quality of the data in the database. And if it's really bad, they may just discard it. Thirdly, a database helps us make sense of the data by providing unique identifiers. Now remember, in our FASTA example, we had the GI and the GB numbers. These are unique IDs for different databases. GB in particular is for GenBank, okay, which is a common database that we'll be looking at as you go through this course. So a database helps us make sense of this data by providing unique IDs for each of the data items. So you can search on this. You can, the next point I'm going to make is especially important for having unique IDs. And these IDs are quoted in research papers. So if you have deposited some data that's very, very key and you've published a research paper, you will usually include the ID of the data that's in the database in your research paper so other people can look at it. This is known as data deposition, depositing of data. It is exactly as it sounds. And in fact, data deposition is now required for you to be able to publish certain papers in bioinformatics. Protein structure is the most common case that I'm used to in particular, where you deposit your protein structure into the protein data bank, which is the main repository, and you sorry about that. You deposit data into the PDB, or the protein data bank, and you get back an ID. And the PDB ID is quoted in the research paper. And a centralized database, what's really good about it is that it allows free and easy access to everybody. So you can go in there and explore structure and sequence and play with the data. Science shouldn't be hidden, and bioinformatics has these databases to make the data easily accessible to people. Some common databases that you will absolutely be visiting in this course: GenBank for sequence data, Protein Data Bank, or PDB, for structures, or WWPDB is the official worldwide PDB, that's the official name for it now, and PubMed for publications. Don't forget, publications are an important part of bioinformatics, and bioinformaticians are often very interested in tying publications together with the source data. So you could have a paper in PubMed that references something in GenBank or PDB, but conversely, if you go to GenBank or PDB, you will find related PubMed publications for a given sequence or structure. So they work together. Now, databases in a networking context. So large data repositories are maintained and mirrored around the world. So this is a term, mirrored, okay? Which means they're duplicated. So the Protein Data Bank, or the PDB, has 3 mirrors. One's in the US, one is in where I am here in the UK, and one is in Japan. Why do you think it's necessary to mirror this kind of data? Think about it. I'm going to give you a few seconds. I'm even going to give you a hint. So remember, as you think about this question later, remember our discussion on network topology and geography. That's the key to answering this question. Okay, so we've looked at databases. Now, what kind of data goes in there, and how big does this data get? I've constantly been saying huge and big and using all these vague terms. So we're going to have a very precise application of this, okay? And this is an exercise for you to do. We have a case study, and we have the human genome. And we're going to estimate the size of the human genome as a text file. Now, this is a common application. You've got some biological data that's stored on disk. These things take space, and you need to know how big these things are to work with them. We're going to store the text file as 1-letter DNA codes. And to solve this problem, this is the information you're going to need. The human genome is approximately 3,300 megabases in size. That is million bases. So 3,300 million bases. That is 3.3 billion bases in size. Each character or letter on the screen and in a file takes 4 bytes of space when it's stored on the disk. So how big is the genome in a plain text file? We're not looking at any metadata or FASTA headers or anything like that, so don't worry about it. Just assume that the entire human genome, 1-letter codes, is dumped into a text file, and you hit save. How big is that file going to be? And it's just an estimate. Now, the reason we went through the exercise is because bioinformaticians and software developers are faced with such problems every single day. So when I face a problem like this and somebody says, do we have the capacity to compute this data? My first question is, how big is the data coming in? And doing some simple calculations like what we just did makes it very easy for you to plan. Maybe you need to buy a new hard disk to store this data. Maybe you need to upgrade your network to allow faster speeds. Having these sorts of simple estimations is very important for you in order to be successful at designing scripts or using bioinformatics software. So one example is the human genome exists. If there is a lab that does bioinformatics, I can pretty much guarantee you that they have a local copy of the human genome in particular, because they're going to work with the human genome a lot. Why are these things kept as local copies instead of downloading them each time? Why do you think this is? Again, we've discussed this. It goes back to some of the networking issues we discussed. So why do you think that is? Now, hopefully you've calculated this number, and it doesn't seem very big. In fact, it seems like you could store this on your laptop. And guess what? Nowadays, your simple laptop, even some tablets and iPads and things, may be able to store this information. That's because disk space is very cheap today. But remember, back in the day, this wasn't the case. Hard disk space was very, very expensive. So nowadays, you might think that these calculations may not be so important. But let me give you another example where this becomes very important. Designing your software and using your software. Let's continue this analysis with the human genome. So we know, based on your calculation from the previous screen, that it's easy to store the genome on disk. Now, let's say you're writing a Python script that needs to analyze the genome. It needs to do something with it, okay? The first step is that your code is going to load the entire text file that we just created of the human genome into memory, or RAM, in order to perform some calculations. Now how cheap is it to manipulate the genome? So think of a case where you have a single computer that's running 10 versions of your script. 10 people are accessing it. There's only one set of RAM in that computer. 10 people are going to be loading the genome into RAM. Does that computer have enough RAM? Can your laptop handle it? So think of this case where you have when you're running this on a single computing machine. And now think, is this a good way to design the script? So you see how our simple experiment, our simple estimation has given us a very good idea for designing the script perhaps a little bit better. I'm going to let you think about that. I'm just going to take a quick sip of water while you think about this. And here is the moral of this slide, and really the moral of this lecture. If you're using software, if you're building software, be very careful with how you write it yourself or how you use them, okay? Memory, or RAM, is very important because it's much faster to work with. So you can store a lot of things on your hard drive, but pretty much anything that works with files on the hard drive is going to need to load it into RAM. So RAM is obviously much more expensive than hard disk space, and also it has other issues that we won't get into, okay? But RAM is basically what you need. As long as a program is running, it's going to need RAM. This is why everyone says upgrade your RAM, so you can do more things simultaneously. So think about ways that other software packages overcome this issue. Now, there are packages that load the human genome in memory, and they can do it on your laptop at home. So just have a thought experiment. It's nothing formal. Just think about ways that other software packages could get around this issue. You don't need to be a computer scientist to answer this question. It's just a very basic survey, okay? Now, finally, I want to conclude by looking at the mindset of the bioinformatician. So we have started our journey at information networks. Then we went through some of the uses. And then we went through understanding bioinformatics data and understanding why the data is big and how big it is, roughly. And we understand now that this puts pressure on our networks, our nodes, our computing. And this is why computing resources are so important for bioinformaticians. Furthermore, designing good software is extremely important in bioinformatics. Okay? So I want to end this lecture by looking at the mindset of the bioinformatician. Let's get started. Remember, you are a bioinformatician in this class. When you're working with information networks based on everything we've learned today, I want you, when you're doing your labs and when you're studying, to keep some things in mind. Number one, bioinformatics is unique, even within the realm of computer science. And it places very special challenges on existing computer hardware and network infrastructure. So remember that your home network, where you can plug in a router and you got a TV connected to it perhaps, you've got your Xbox or your PlayStation, that home router and that network is not designed primarily with transferring huge amounts of files. Huge amounts of files. I'm talking genomes and bigger. So keep that in mind. This is where the software design comes into play. If you are writing scripts, or if you're building software, design them very carefully with the data sizes in mind. This is the best thing you can do for yourself. Before you start plugging away, writing scripts or software or downloading human genomes left, right, and center, think about your networking. Think about the challenges. Think about why a file is taking a long time to download. Think about why a computer is taking particularly long to work on a file. Think about what you can do to a particular bioinformatics software to make it run a little bit faster. Speed is critical in bioinformatics. We have very powerful computers. Computing is cheap now. We need to extract the best possible performance out of them. For the consumer, there may not be a big difference between the Intel i5 and the i7 processor. But to a bioinformatician, that makes a world of difference. Because for us, we are operating at the very limit of how fast that computer can go. So keep that in mind. And if you're writing your own scripts, be nice. Understand what your computer is capable of doing and build your software with your hardware limitations in mind. Now, again, I don't want you to think you need to be a hardcore computer scientist to do any of these things. Most bioinformaticians are actually biologists that have learned a little bit of Python scripting, Perl scripting, and they go away and they do remarkable work. And one of the things you're going to pick up as a bioinformatician is the ability to write software. Okay? It just is going to happen. So don't worry. You're going to pick up these skills. My job as a bioinformatician from a computational point of view is to encourage you to think of the design decisions before simply just plugging in and writing a script. Network, databases, they have mirrors. Use them. As I just mentioned, the Protein Data Bank has 3 mirrors in the US, the UK, and Japan. Did you ever think about why this is the case? No, it's not just politics. It's a very fundamental reason. If you're in the US, you will use the mirror closest to you. Remember what we said about network geography and topology. If possible, use the network that is the closest to you, because chances are that's going to be the fastest. Now, maybe you get lucky and you can tap into the Japanese server when the Japanese scientists are asleep and nobody's using their network. And maybe that will be faster for you. But in general, use network topology and geography to your advantage. Oops. Now, this is something I haven't really touched upon, and that's for good reason, but I want to put it on here because it is very important for the mindset of a bioinformatician, and to some extent to networking as a whole. If you've got various components working together, the speed of the overall process or the network or the pipeline, whatever you're working on, is severely limited by the slowest step. Think of a traffic jam. Sometimes traffic jams happen because there is a tractor in the way, or there's a very slow lorry in the way, or maybe there's a garbage truck that's chugging along and it's stopping all the cars behind it. If you have a Ferrari on a highway and it's stuck behind a garbage truck, that Ferrari is going to be moving as slow as the garbage truck is. It doesn't matter that it's a Ferrari at that stage. Same thing with networks, same thing with pipelines. They are restricted by the slowest and usually the oldest software component. So if you are designing a pipeline or if you're using a software package, remember pipelines can be single software packages as well. If it's particularly slow and you don't know why, look for upgrades. Perhaps there's a newer version that's resolved a lot of these issues. This actually brings me to my next 2 points quite nicely. Before starting a bioinformatics project, just like the software design case, make sure that your simple things, these are things that people will always make mistakes on, because you just don't think about your disk space and your RAM every day. But make sure you have enough. Do some simple estimations like we did before, okay? And of course, in your labs, I'm going to try to give you an example that you can work on in a tutorial or in lab as we go forward, where you get to do some of these simple network estimations. It is just a very, very useful mindset to have, and it just saves so much grief when the time comes and somebody is asking you why something isn't working. You can go back and visit your estimations, okay? And hey, if you've done your estimates right, chances are nobody is going to ask you that question. Now, on the point of the slow software, bioinformatics is also unique in that the software is generally community-driven, and most software packages are free. And when I say free, I don't mean free as in, there's a phrase. It's not free as in beer, not free as in free beer. It's free as in freedom. Free software, of which open source is a derivative. And we can discuss this at great length later. This is a topic that's very interesting to me personally. But basically, it encourages collaborative development of software. The plus side for you as a bioinformatician and for you in academia and in research is that these packages get updated very frequently. And sometimes you will build a beautiful bioinformatics pipeline that does something really well, and then you need to go in and upgrade a particular piece of software because it does something even better. So keep that in mind. When you design bioinformatics pipelines, when you use bioinformatics pipelines, look at the version numbers. See how outdated it is, because these packages move quickly. And sometimes it's in your best interest to use the most up-to-date version of software. Okay? Now, there are cases in high-throughput facilities, like we have some here in Oxford, where some of the data sets coming out of some of the older machines are limited by a certain file format or a version, and they are reliant on a particular version of a software tool. In those cases, it can't be helped, but that is generally not a good design. If you have control over designing your pipelines, make sure you look at the version numbers and pick versions. Generally, the newest ones will be the best. Now, this concludes my presentation. Before I let you go, I just want to reiterate a couple of things. You are in this course because you are interested in bioinformatics. You're interested, maybe you know what the term vaguely means. Maybe you have a good working knowledge of what bioinformatics is. But in all cases, my experience working with other bioinformaticians is that they may be excellent biologists, but they don't make these simple design decisions when it comes to software. A computer is a finite entity. It has a certain amount of space. It has a certain amount of power. That's not to say it's weak. Computers are extremely powerful. And we, in this course, at least as part of my lectures and my modules, I am going to try to encourage you to think about efficiency in using your networks effectively, in using your pipelines effectively, if you're writing scripts, not to make silly mistakes that may add 2 extra days to your processing. This is a true story. I was working with an algorithm that had a simple software implementation. Now, thankfully, the software was free software, so I could look at the source code. But very simply, it was doing a repeated action. And just by accident, the original developer had copy-pasted a line twice. The same line did the same thing, but it has now doubled the speed of the program running. Simply by deleting that, I brought the processing down by half. Simple things. Now, that was a mistake. It wasn't a design decision. But you can think of cases where the design decision itself has not been cleverly thought out. So in this course, I'm going to try to get you to think about the limits of what we can achieve as bioinformaticians from the computing resources that we have. Okay, so keep that in mind. And I will have some questions, and I will be able to take some questions during the tutorials, and I believe I'm with you for some labs as well. So I'm going to try to design my modules around some of the lecture goals that we talked about today. So I hope you enjoyed this. I hope it was not too confusing or difficult or any of those things. If it was, please drop me a line. Remember, this is a course that we're trying to get off the ground. It is an exciting science, and we need your input and your participation as much as possible to make this course a success. So I really hope to hear from you soon. And yeah, thanks for listening. I hope you enjoyed.